Welcome everybody to the NextGen Microfluidics webinar in a series of final dissemination webinars from our project. Today's webinar is foil-based microfluidic chips for multiplex in vitro diagnostics. Is this a game changer for lab quality at low cost? So we're really happy to have two presenters from UNAM Research, Christian Wolf, and from GenSpeed Biotech, Max Solleitner. We're all here together in Austria this time. We've had other international partners at our previous webinars. I'm Caitlin Ahern from BioNanoNet based in Graz, and Christian Wolf at UNAM Research and our coordinator, Martin Smolka, are here from also in Graz, and Max Solleitner from GenSpeed in Linz. So our coordinator, Martin Smolka, will be giving a short overview of the project, and then we'll launch into the presentation. So we'll start with GenSpeed and then go on to UNAM Research. At the end, we'll have a Q&A session, but of course, if you have any burning questions, you're welcome to put them in the Q&A, and we'll remember to call on you at the end. So I'll hand over to Martin.
Yeah. So when we started the project NextGen Microfluidics, we saw a big challenge in microfluidics in general, and that is that development of chips, development of microfluidic innovations is complex. It typically includes several solution providers which develop different steps of your device. So if you have a new microfluidic innovation and want to make a product out of it, you need to talk to multiple players which implement different steps of the development. And yeah, that's a big challenge for the customers. So they need to choose different partners, need to ensure that their technologies match. And with this challenge in mind, we did the following. So first of all, sorry, here you see the logos. First of all, we acquired quite a lot of funding for the project. It's a rather big EU project, and we have all in all 21 partners. And we chose them since they are solution providers for development and production of microfluidic devices, and they're at different positions of the entire value chain, starting from simulation to final production of components. We founded our own startup. That's the Microfluidics Innovation Hub, in short, the MIH. And the MIH bundles all our services as a single entry point. So now customers can address just one entity, that's the MIH. And the MIH bundles an offer with combined solutions from all of us. And during the project, we did already verify our portfolio of combined services. So we had a few demonstration cases. A few of them were known from the beginning. They followed a concrete work program, which we knew from the start. And then with these first references, we also went out to the market, and we asked for external customers which have specific challenges to be solved. And we worked on these challenges in our open call program. So with that, we have already 22 applications in which we demonstrated successfully that we can offer our combined service portfolio and can solve problems in microfluidic chip development, microfluidic solution development. So as a summary, we as the Microfluidics Innovation Hub, we are contributing to the microfluidic revolution with cutting-edge technology, and we cover a broad range of solutions along the value chain. So first, all about the chips and the cartridges which you need for the microfluidic technology. So with manufacturing of these consumables, and then also with integration of functionality, sensors, biosensors. We have expertise ranging from chemical, biochemical to biological processes, so that we can cover everything, all microfluidic chip which do measurements, which are used for in-vitro diagnostics, point-of-care diagnostics. So all kind of lab on a chip applications, but also organ-on-a-chip applications where you grow cells in defined environment and study their behavior inside of microfluidic chips. So just contact us for your microfluidic development just by approaching the Microfluidics Innovation Hub. That's the most important about the project, and we are also ending the project with a small conference related to microfluidics technology that is here in Graz in Austria on the 26th of March. So you can also meet us in person on one day dedicated to microfluidics. So we are happy to see you there in 2 months. And with that, you see an overview of all the partners, and I will hand over to Max.
Okay, thanks, Martin. So I'll start sharing my screen. So hello, everyone. I have the honor to introduce you to our microfluidic chip-based multiplex microanalyzer technology. So today we will cover the topic of the foil-based microfluidic chips for multiplex in vitro diagnostics and ask the question if this is a game changer for lab quality at low cost. So we did a lot of work in that topic, let's say, last 10 years even. So let's get into it. First of all, I want to introduce you to the technology, and after that, Christian will take over and tell you how we approach the foil-based chip approach. So I want to briefly introduce you to Chainspeed, the company I'm the CEO of. We founded the company as a spin-out of Greiner Biovon, which you may know. They are famous for blood sampling in Europe. And our focus is development, production, distribution of rapid tests for really different applications. We have the basis technology. This is the Chainspeed platform technology. So maybe you see me, and then you also see our device. So that's the platform technology everything is based on. And we develop tests based on the technology, which are typically IVD or CE certified. And now we are also IVDR certified, which was a big step for us. So we have different product areas and a two-way strategy, so to speak. So one strategy is that we offer our technology to OEM partners. Could be you, for example, when you have lab tests, for example, on the market, you want to move them to point of care testing, then we can easily transfer that for you onto our technology. And of course, we have our Chainspeed branded products, which we call the Xbox for multiplex and point of care tests. And there we have developed different tests. I will guide you through those tests in a minute. So how does it work? So we have a microfluidic test chip, and this will be the topic of that talk afterwards also. So you can see it's pretty simple. It's a straight channel with capture molecules that goes into the device, and the device is closed. The test is running, and everything is controlled and navigated by the software that we developed. So this is the setting that you have, for example, at the pharmacy or the doctor's office. You can see the device that is connected via USB and also power supplied via USB by a tablet computer. And this is a typical test kit that we provide, including everything, test chips and all the pre-analytics tools that you need. And now you see an animation that briefly shows how this works. So, of course, everything is automated. It's a short turnaround time, about 20 minutes. We can detect up to 8 different analytes in one test run and quantify them. And the system is easily adaptable to different applications. So you put in the test chip, and then there is a system preparation procedure or not, depends on the assay a little bit. Then you add your sample. Typically, it's done by a dropper bottle in point of care and not using a pipette. Then the chip goes in, and now you see how this works. So there is a focus now on one of the capture molecule bands that we have in the microfluidic channel. And then when you know an ELISA test in the lab, it's typically it's exactly the same. So the antigen or another molecule binds to the antibody, And then what you can see here, the fluids are injected automatically into the system. In this case, we have a sandwich ELISA, so you have your bioattenuated capture and detection antibody that also unspecifically binds, of course. Then with a washing procedure that washes away the unspecifically bound antibodies, and then we generate chemo-luminescent signal in the different bands with different intensities. And then we do a data analysis in the software, and you end up with a PDF report on the tablet. So that's how it works. And this is the advantages that we have. So we combine the accelerated hybridization reaction using the microfluidic structure, because there are capillary and shear forces. So there is much higher probability for the biomolecules to bind and interact with each other than with an ELISA in your ELISA pot in the instrument, for example. And we combine that with very sensitive chemoluminescence-based optical detection system, and also with an automated dispensing system of the reagents using piezo membrane pumps. And this all ends up in a multiplex microELISA technology. So it's fully automated quantification up to 8, and with a different chip type, even 16 parameters that you can quantify in parallel in 15 to 25 minutes from various sample types. So this in summary for the specifications. So it's fast. We can detect up to 8 different analytes. We can also do RNA-DNA combination. It's not the priority topic today. It's very sensitive, as you will see, picograms per mil. Sensitivity is not a problem, and IVD CE certified. So the benefit of the system is that you can really put any ELISA system on the test chip, combine it with 7 others, for example, and do a microELISA. And even we can combine, for example, an indirect ELISA with a competitive ELISA for only one target. So that means you can broaden your dynamic range. As mentioned, you can also use that for DNA, RNA-based assays, as we do, for example, for a periodontitis test. So in this slide, I only want to figure out or emphasize that the sensitivity is pretty high, so we can go down to the picograms per mil. This is one of our current OEM examples where we use a sample from synovial fluid from hips or knees to detect 4 different biomarkers in one test run. So that really means and quantify it. So you see here the calibration curves. For example, biomarker 2 has a clinically relevant range is indicated in yellow here, and the calibration curve is here. So that means all of the 4 analytes can be quantified within 25 minutes with lab quality. And this is really a quantum leap here. So compared to strip tests, they have nice handling, nice usability, but often they are problematic with regards to quality. Lab-ELISA is exactly the other direction, so it's nice quality, but not so nice usability, and the chain speed provides the best parts of the 2 worlds, including the multiplex capability. So this is one example. So just figuring out or emphasizing that we can do OEM developments was a company where we put the supernostic test onto a point-of-care device. Then, as mentioned, we can do also DNA, RNA-based testing. So this is pathogens for periodontitis. And then we're coming to the protein tests. So this is immunoassays. This is a vitamin D test that we developed. And typically you see what we have on our microfluidic chip. So we have control lines there. And of course, the specific vitamin D in this case. And what's important for you to know is how this works. So this is how it works at the pharmacy or doctor's offices. So you open up your test chip pouch, then you take out the test chip, then you disinfect the fingertip. You take 20 to 50 microliters of full blood, and this is all off-the-shelf components that we use there. Then we put that in a small vial dilute it to the right dilution. And then we have developed a small filter tip that goes on top, and that we use to do the blood serum separation. So if you look now, when we apply the drop to the test chip, then this is transparent. And then the chip goes in, and depending on the assay type, there's another step where you add manually the detection antibodies. That are biotinulated or not, depends on really on the assay. So sometimes it's only one step and sometimes it's two. In this case, it's two. And then you see the software in the end gives you the report. So this is how it works. And this is how, for example, we return your measurement or provide a reporting. So you can see here the concentration of vitamin D in the full blood is 70.6 nanograms per ml. And this is a nice illustration. Then we developed tick-borne encephalitis, so it's really nice for detecting antibodies system. There we did multiplexing, so tick-borne encephalitis antibodies and yellow fever. So the system is really good for multiplexing. That's why it's called XPOC testing. And again, you see the other controls. So we of course, we always do the clinical studies, so we get really, really nice values for the sensitivity and specificity in comparison with the lab systems that are out there. And this, for example, is a lab report. This is a semi-quantitative output because that's enough for tick-borne encephalitis. So the system tells you if it's necessary to have a vaccine boost or not. That's examples that we developed. And the most important example, of course, was the COVID-19 antibody test that we developed. It's a multiplex test also. And that was also let's say the focus of the project of the next generation microfluidics project was to put that one into a foil chip, and you will see why we did that. So this is now the test. As you know, the COVID-19, the SARS-CoV virus looks like that. So we have the spike proteins here. Then we put two bands here, the full S protein and the receptor binding protein. But in addition, we put antigen of the nucleocapsid protein there, which means when you test your blood, you will see if there are neutralizing antibodies, then you get a signal at the full spike protein or the receptor binding domain. And when you had a natural infection with the virus within the last 6 or 12 months, you also get a signal here. So that allows to discriminate between vaccinated people and people that had also antibodies from a natural infection. So this is the result here. You get a concentration, and for the neutralizing antibodies, so that means you know if the protection is still valid or not. And in the second line, you get an information if you had a natural infection within the last 6 or 12 months. So this is also interesting for people that suffer from long COVID, because you can detect if there was an infection. So what's also important is, of course, that regarding the cutoff level of our system, so we are really comparable to the lab systems. So this is our 10 binding antibody units per ml is our limited detection, and if you compare it to the big ones, Abbott, for example, is at 7, and Diazorin is even worse than ours. So it's really comparable to lab systems. So this is what we did in 2021. So we sold. At the moment, we have around 150 ChanSpeed R2 analyzers in Austria, mostly at pharmacies, but also at doctors' offices. So we sold more. In sum, we sold more than 100,000 antibody tests. And then we came up with the question, of course, so is it scalable? What about the costs? And of course, we had to compare our system with strip tests because they're out there. And as you know, they flooded whole Europe. So the antibody test in the beginning of the pandemic flooded whole Europe, and basically they came from China. So how does the ChanSpeed system compare with strip tests regarding the key performance parameters? So the usability, I would say it's more or less the same. So we have one step. In best case, they have one step, so that's fine. Turnaround time, ChanSpeed is maybe a little, that takes a little longer, but it's lab quality in the end. And we have at least 10 times better limit of detection. So as you've seen for the COVID-19 antibody test, there we have a much better limit. So that's very important, of course. Then we can quantify very well compared to strip tests, and we are robust, of course, in comparison. And what's important to mention is that with a strip test, typically you have problems with the multiplexing because you have depletion on your nitrocellulose, and that's very much better. So we can really have up to 8 targets quantified in one test run. But, and that's the key question also in this webinar, the challenge is, of course, if you have a microfluidic-based diagnostic system, then the costs are higher typically than costs of a strip test. So the goal is, of course, to be comparable in that direction. What's the principal advantage of strip tests? I think we can all agree that's the low material costs, and of course, I think the highest advantage or the biggest advantage is, of course, you can manufacture them in a roll-to-roll production manner, and this is how they typically look like. So this is from Biodot. So you have your nitrocellulose here, your roll, and in the end you have, in the best case, you have your strip ready. So this is what we are doing. So we have, for our chain speed test production, typically the standard way. So we have our plastic foil, and there we do immobilization of the capture molecules with a microarray printer, which is pretty complicated, of course. And then we add an injection molded top part that is also that has also the microfluidic structures on top, and then we bond that together using ultrasonic welding, and then we end up with the chip, with the inlet, and this is watertight, of course, with the reaction channel where all the things are happening, and with the waste reservoir. And this is capillary based driven, so everything that goes into the inlet is sucked through the channel into the waste reservoir. So, and the costs of good for this chest chip, typically in this production process, is around €1. And to be compatible, fully compatible with strip tests, of course, the goal is to get much less. So much below €1. And then the question is, or was when we started the project, can we do that in a roll-to-roll process? And now it's my honor to hand over to Christian, who will tell you if this came true or not, or where we stand. Thank you.
Okay, it's my honor to present you and give you a little bit more in-depth detail concerning the technical aspects of our demo case development. So especially, as Max already said, the development of a high volume production process for those microfluidic chips used in the chain speed device. Okay, so very shortly, because Max already said a lot about that, what was the overall aim of our demo case? So Max already showed the impressive parameters and properties of their point-of-care diagnostic system, which can give very reliable results, for example, concerning your COVID-19 antibody status within 15 minutes point of care. So that's also the test and the application we were focusing on in our developments in the demo case. And as we learned, I would say that the crucial component of this device is this microfluidic chip here. Yeah? So it's a disposable product, so it has to be replaced after each test, and the size of it is approximately of a microscope slide, so 7.5 to 2.5 centimeters. Yeah, and as Max showed, up to now, this chip is produced in a rather complex process, in a multi-step process by injection molding and bonding. And now, the idea of our demo case was, the overall aim, to realize this injection molded chip in a fully fold-based design. So something like here in the lower left corner, by means of a high volume roll-to-roll imprinting technology. And as Max said, why do we want to do that? On the one hand, to establish a process which allows for a high volume production of those disposable chips, where potentially a high number of those chips is needed. And as usual, yeah, the most important reason probably is to reduce the cost of those disposable chips. So we were talking quite a lot about roll-to-roll imprinting, but what is this exactly? And now I would like to show you a short animation of the roll-to-roll imprinting pilot line we have here at Yoneum Research, for example. Maybe I will disable my camera because I hope that the video is then more fluent. Okay, so everything starts, so it's a little bit loud. Everything starts with surprisingly with a big roll of a substrate foil, a carrier foil. So in our case can be with a width of up to 280 millimeters. Material-wise, you're quite free, but it's the only important thing is that the foil is transparent. Why? We will learn later on. So then there's a cleaning step, and you can also do some corona pre-treatment, which can be important for the next step to improve wetting properties, for example, because then this carrier foil is coated with a thin layer of a resin, an acrylic resin, which can be cured and hardened by UV light. And this thin film is then transported between two rolls, where one has a structured stamp on one side. And then you can press these structures on your roll into the still liquid resin film. And while the resin and the foil is still on the roll, they are cross-linked and hardened from the backside by UV light. And that's also why it's important that the foil is transparent. And after half a turn, the resin is fully hardened and you can demold your structures from the roll, as you can see here in this animation. And there are several applications where you can use that. You can go down to nanoscale with your structures here. So we can use some optical structures like free from micro lenses. And what we did in our demo case, you can also realize microfluidic chips for medical devices in a roll-to-roll process. What's also very important, of course, especially for medical applications, that you have a full inline quality control. So we installed a video camera so you can check every single chip concerning potential defects or deteriorations whatsoever. Yeah, and finally, you can then upwind your structured foil again to a big roll with your samples. Okay, so that's one core process of this full realization of the chips, but it's not by far not the only one. Because in order to really realize this commercial injection molded chain speed chip in roll-to-roll mode, you have to address way more different process steps, which you can see here in the workflow chart diagram of the demo case. So everything starts, for example, with material development. This is for example, tuning of the resin. So this UV needle resin we are using, so that can be tuned, for example, concerning contact angle. As we learned from Max in the chain speed device, there are no bumps involved. Everything happens by capillary forces alone. So therefore, for example, the contact angle is very important, of course. Then in the next step, you have to adapt your design to the limitations of the roll-to-roll imprinting process. We will see that in more detail later on. You also need a roll-to-roll master production, or as we call it in the language, in the roll-to-roll language, shim. So these are the stamps which are wrapped around the actual cylinders, which press the structures into your resin film. The next steps is the actual imprinting. You've already seen the animation of the UV needle imprinting process, but in our project we also developed a second, quite similar process called extrusion coating. I will show that in a little bit more details later on. Yeah, so after the imprinting, you still have no finished chip, because then these are just channel structures on a carrier foil. So you still have to introduce some biomarkers, biomolecules sensing spots, and that's typically done by a spotting process, which was also developed for full roll-to-roll mode. And then, of course, you still have to close your chips. Yeah, they're still open. So for the fluidic to work, you have to close it you have to bond it with a cover foil. And here also the two imprinting processes, they ask for two distinctive bonding methodologies. And finally, you still have the back-end processes. That's mainly inlet cutting, singulation of the chips up to packaging. So the cutting processes were done by laser cutting in our demo case, which showed to be the best suited methodology. Okay, and now I would like to have a closer look to some of those processes. For example, let's start with this design step here. As I already said, you have to adapt your design to the roll-to-roll limitations and boundary conditions. When you look here, so that's the design of the commercial injection molded change speed chip, what it looks like at the moment. And here on the left-hand side, you can see the final design of our roll-to-roll imprinted chips. And you can see there's quite a big difference. And that's mainly because for two limitations of the roll-to-roll imprinting process, one of it is that we are limited concerning the imprinting depth. So that's typically limited to 200 micrometers, something like that. And when you look at the change speed chip here, the compartment, the waste compartment has a depth of 600 micrometer. That's not doable by roll-to-roll imprinting, because you still need a substrate foil, you need an cover foil, so that easily sums up to something like the thickness of the full chip of 900 micrometer to 1 millimeters, and that would be no lap on a foil anymore, so it's then probably more lap on a plate or something like that, where you also already get problems with upwinding and so on. So that's one limitation you have to consider in your design, and that's why also the roll-to-roll chips have those big waste channels, waste compartments, because they are needed to take up all the liquids which are used in the testing protocols on the chip. And as a second limitation of the roll-to-roll process, which you have to consider in the design, is the design of fittings, meaning if you have inlet structures, like on the injection molded chips, 3D inlet structures or outlet structures. So that's very difficult to realize in roll-to-roll. So you have to come up with some sort of two-dimensional solutions, but you can see here, there we have, instead of the spotlight structure, we have a 2D inlet in this elliptic shape. Yeah, typically these designs, things are supported by computer simulations here in-house, where you can simulate the fluid flow, for example, but you can also have a look at more sophisticated problems. For example, what you can see here, that's a simplified model of the sensing channel of the chip. So maybe you can see it a little bit better in the next slide. Yeah. So that's typically it's a cross section of the sensing chip, as it is, for example, in the change speed chip, and maybe you have some sort of deterioration at the bottom where the contact angle is different than the surrounding matrix material, or you have a sensing spot which another surface energy. And with the simulations, you can, for example, check or investigate what happens when you fill that by capillary forces. And you can see here, for example, the result of such a simulation. And if you have such a spot with a very high contact angle at some point, it can happen that an air bubble is formed there. And that's something, of course, you want to prevent in any case. But with the help of the simulation, you can investigate that and come up with some workarounds, like changing the contact angle of the sensing layer or do some geometric adaptations to that. Okay. Yeah, so maybe let's have a closer look to the core processes in the chip production, and that's the UV anneal imprinting. So you have already seen a quite nice animation of that. But here maybe you can see a video of the real imprinting process. So after optimization, that's how those chips, the Demo 2 chips, run out of our roll-to-roll imprinting device. And as I said, one chip, we have 6 chips in a row. Maybe you can even go higher than that. And one chip is 7.5 centimeters. and the web moves with approximately 2 meters per minute. So this sums up that you can produce around 8,000 chips per hour. So that's a really, I would say, a high volume process. So the daily production with 2 8-hour shifts then sums up to something like around 130,000 chips. So that could be established very successfully. And here on the right-hand side, you can see a real image of this roll-to-roll pilot line installed at UNAM Research. Yeah, and as I told today, that's not the only imprinting process which we developed during the demo case. It was also a second methodology developed mainly by the company partner InMold. That was the so-called extrusion coating. It's a somewhat similar process, but the difference here is that you don't use any UV curing resins, but you use a thermoplastic polymer material instead, so something that gets soft and when you heat it up. And there you also apply a thin layer of your melted polymer that's melted in an extruder and then extruded with a broad slit onto this carrier foil. And then you directly press your structures while it's still warm and soft into this polymeric material. And while the roll moves, the thermoplastic material cools down and hardens, and then you have your structures, the structured foil and the structured microchannels whatsoever realized on this carrier foil. So for example, here on this side, you can see a video of the demo chips, which were then produced with this extrusion coating material. So there you have some kind of thermoplastic polymer as a matrix material and not an acrylic resin, polystyrene or something like that. Yeah, and then, as I said, after imprinting, you still have no working chip, of course. Yeah, you have no chemistry in there, no sensing spots. And also the channels are still open, and it was something that was addressed also in roll-to-roll mode. So here you can see the principle. The spotting is done with automated dispensing of the catching molecules into the sensing channels, and then the chips are closed with a cover foil in order to enable the actual spotting process at the fluidic, so that the fluidic works. And also have a short video showing that process here. So up to now, it's still in a stop and go mode. The spotting is done, but that can be further improved in a continuous process. But we can see here, so the web is transported into the dispenser, and after a short outer alignment, you can see how the biomolecules, the chemistry, the markers are spotted into the channel automatically. So we are just using 4 nozzles for this process. This can be scaled up, of course, when you're using more nozzles in parallel. So after spotting and a quick quality control, the web moves on to the next sheet, and then the next chips can be spotted. And here you can see a short video of the roll-to-roll lamination process. So where you close the chips with a cover foil. So maybe it's a little bit hard to see. That's the cover foil, and that's how it is then connected with the actual chip, the imprinted and spotted chip. With the UV anneal process, imprinting process, this is done that on the cover foil, you also have a very thin layer of this cross-linkable resin, and then the imprinted chip and the cover foil are pressed together, and then you expose it with a small dosage of UV light again, and that's how a very firm bonding can be realized. This cannot be done for the extrusion coated process, of course, because there is no cross-linking UV resin involved. There was the bonding was developed and established similar to the chips from Chainspeed by an ultrasonic welding process. Okay, and what you can see here, that's a direct comparison on the right-hand side in this picture. That's the injection molded commercial chip from Chainspeed. And in direct comparison to that, you have one of our roll-to-roll imprinted, spotted, and bonded chips. So as I said, they look quite different. What we also did here on the right-hand side is that we did a thorough testing of the chips concerning fluid flow, leakage, and so on. And after the optimization of all processes, very minor leakages and samples not working concerning fluid flow and so on could be observed. So typically this is already done before singulation in these tests because there's no need to do singulate the chips by laser cutting before. Yeah, and as a final evaluation of the roll-to-roll imprinting process, we tested 200 randomly selected chips concerning their signal generation. So that will be a direct comparison between the commercial injection molded chips and the roll-to-roll chips concerning the signal height sensitivity of the chips using the COVID-19 antibody detection assay from GenSpeed. So that was already shown by Max. So typically you have, this is the sensing channel. So that's similar or the same as with the injection molded chip on the roll-to-roll chip. And you have these 4 sensing areas where you detect 3 of them are for the proteins, for the antibody proteins of the virus, and 1 is a positive control. And then via a chemo-luminescent reaction, you get 4 distinctive light signals, which can then directly compared between the chips to test their performance. And as I said, we produced quite a high number of chips, but yeah, that's quickly done in the imprinting process. As you remember, when you're producing 8,000 chips per hour. So we selected 200 of those chips and yeah, compared that with a reference sample from GenSpeed concerning their performance. And that can be seen here. So these are the signals of those 4 sensing spots. For in red, these are always the roll-to-roll imprinted chips, and in direct comparison to the blue ones, these are the commercial GenSpeed chips available on the market. For juvenile chips, we did this for the juvenile imprinted chips as well as for the extrusion coated chips. And as you can see here, in direct comparison, in summary, you can say that the roll-to-roll chips performance is in the same order of magnitude than the commercial chips concerning a signal height and signal quality. Some a little bit higher, some a bit lower, but overall, I would say, yeah, the signal performance is comparable, which was, of course, a nice success of our demo case. Yeah, and that also already brings me to a short summary. So I think we could show in the demo case that the full roll-to-roll production of microfluidic chips is possible, and that it's really a high volume roll-to-roll production of those chips. And 2 distinctive pilot lines were established. So one for this juvenile imprinting, just maybe juvenile means UV light cured resin, and nil is nano imprint lithography, because as I said, you can go down to nanoscale of your structures. That was not needed in the demo case, but it's possible. Yeah, and the performance of those roll-to-roll demo chips are comparable to the commercial products. And then as a final summary, we made a nice 2 minutes promotion video of the demo case and the chip, and I think that really nicely sums up all the developments we had so far, and that's what I would like to show here.
Maximum diagnostic relevance from minimal sample volumes. This key feature of microfluidic lab-on-a-chip systems will set the future standards of medical diagnostics. Often based on disposable setups, the demand for a low-cost, high-volume production of the chips is continuously rising. Modern roll-to-roll patterning methods meet all requirements for fast and cheap production of lab-on-chips in one continuous process. In cooperation with GenSpeed Biotech and partners from the Microfluidic Innovation Hub, low-cost roll-to-roll pattern chips are developed for COVID-19 antibody tests in GenSpeed point-of-care devices. All necessary steps are addressed by the partners over the full production chain. Development of basic materials including biodegradable approaches, chip design and fluid simulations, mastering for prototyping as well as for large-scale production, patterning by UV nano-imprint lithography or by extrusion coating, surface functionalization and bioanalyte spotting, back-end processes including bonding, cutting and singulation. All steps accompanied by quality control measures to highest standards. Microfluidics Innovation Hub, we get microfluidics rolling.
Okay, so thank you. And with that, I would like to hand over to Caitlin again, I think.
Great, thank you so much, Christian, and for your presentations. I will share the screen briefly. If you're, in addition to the demo case that was presented today, there were 4 other demo cases that we've been presenting in these final webinars, and we had 18 open call cases that took advantage of services from the NextGen Microfluidics partners. Our project is coming to an end, but all of these services will be offered by the Microfluidics Innovation Hub. Again, the spin-off that Martin presented at the beginning. So if you're interested in taking advantage of these services, please check out microfluidicshub.eu. I also put the link in the chat, and you can find out more. So now we have a couple minutes for questions. I also put the email addresses, contact info of our 2 speakers here. So please take them down if you have individual questions. But if anybody from the audience has a question, feel free to raise your hand, unmute, and ask it yourself, or you can also write in the Q&A. But in the meantime, as people are thinking, I would have a question. So for Christian, we'll start with you. You mentioned the computer simulation for the design. What else can you use this for?
Yes, typically we use these computer simulations for the design adaptations. Typically the laminar fluid flow which happens in the in the microfluidic chips, so for the actual design size and so on. But you can do even more with the simulation. So you can go up to, you can also simulate, for example, chemical reactions or mixing procedures in a chip. That's also always difficult stuff, in laminar flow to get some liquids getting mixed. But it's, a lot of things are doable. Even you can even simulate the sensing performance of electrochemical sensors, for example, including chemical reactions and enzymatic reactions up to you get a signal, let's say, for a lactate sensor, direct function of the sensing signal as a function of the lactate concentrate in your sample. So that's all doable with simulation.
Okay, thanks a lot. Turning to Max, are there other products that are coming soon that are using the GenSpeed technology, if you're allowed to share?
Yeah, sure. So thanks for the question. We have different areas of application. Typically we try to find OEM partners in different market niches where we work together then with them, and we have, as I briefly mentioned in my talk, one which I'm really proud of is in the orthopedic area, where we're starting the clinical studies next week. So it's a 4-plex test. It's really nice. And we hope with the new IVDR in place that we can have that on the market within, I don't know exactly, between 9 and 36 months. So it's really hard to get it through. And there are others. Environmental testing there. We are developing something, and also one of the outcomes of the NGM project in the hematology area is very interesting, and we partner with one of those customers there to get another product on the market, also used in clinics then. So there's a lot to come, hopefully.
Great, that sounds exciting. Okay, any other questions from the audience? Feel free to raise your hand. If not, then I will let you know about our final webinar. We're just in the planning. It will be on March 20th, probably. We'll send out information on our website and our LinkedIn channel, so please follow us. It will be on smartphone-enabled home diagnostics for potassium and blood. And this is the final demo case that we're presenting. So the rest will, if you missed the other webinars, we'll have recordings coming up on our website, so please visit nextgenmicrofluidics.eu. So thank you everybody for coming, and thank you again to our speakers for presenting this exciting work from our project. All the best, and we hope to see you at the next time.